Okay. Um, so last talk of the day, stick with me. It's going to be short. So I'm Carla, and I'm going to be telling you about a tool to decompose bulk RNA-seq data based on copy number. Oh, actually, I need this. Okay, so as you know, tumors are complex, heterogeneous tissue, and when we studied its expression at the bulk sequencing level, this is confounded by the copy number and the purity of the sample. So there's a couple of tools that are able to decompose the actual expression profiles of the cells in your sample, so the different cell compartments, for instance, CyberSort X or DMixT. However, these tools often rely on cell type specific matrices from single-cell RNA-seq data, or the use of matched normal samples. So here we're presenting CREDAC, which is a tool that is based on the copy number. So we integrate allele-specific copy number and expression, and it's a reference-free tool. So basically, we don't need any normal samples or reference matrices or any prior knowledge, but we require the existence of paired DNA and RNA-seq data, which is available for most of the large pan-cancer cohorts such as TCGA or PCOG. So CREDAC is based on two main principles. The one would be that we can explain the expression of any given gene at the bulk level as the expression that comes from the tumor and the normal cells. And this can be further explained as the expression per allele copy, so per unit of DNA, weighted by the purity. So for instance, the proportion of cells of tumor or normal cells weighted by the copy number. So how many copies do you have of that gene? So that would be the first principle. And the second assumption would be that in each cell population, all copies are equally expressed. So imagine we have a situation where a tumor cell, for instance, is a 2+1, so we've got two alleles of one of the maternal and one of the paternal, for instance. We would expect that if this assumption holds true, at the RNA-seq level, we would see also that the green allele, for instance, presents as many, like twice as many copies or counts as the pink allele. So we do not make any assumptions on what is the effect that copy number has on the expression, but we make an assumption of what should be the relationship of one to the other allele at the RNA-seq level. So with that, and at regions of heterozygous NIPS and where we have copy number imbalance, we can actually decompose what is the expression per copy in the tumor and the normal cell. So that is ET and EN. So with that, we apply CREDAC to the TRACERx lung cancer study, which is a multi-region study for which we have matched whole exome and RNA-seq data. And the first thing that we set out to do was to validate this assumption of equal allelic expression, how often it holds true. So for that, we looked at the heterozygous NIPS, and we counted the number of reads in alternate and reference alleles, and then counted the percentage of allele-specific expressed genes, so how often we found genes for which this assumption doesn't hold true. And you can see here this is less than 10% in most of the cases. So we can be quite confident that our assumption would hold true in most of the cases, but there's going to be some genes for which our tool will break. So this is just some example of the type of output that you would get out of CREDAC applied to TRACERx. So for instance, here we have just a list of genes, and now each dot represents a SNP for which it had power to deconvolve. So based on these ET and EN estimates, so the expression per copy in the tumor normal cells, we can then estimate the total fraction of the tumor expression. One of the main limitations or challenges that we have with this tool is that we're unable to deconvolve all the genes of all the samples. So we end up with a matrix like this one, where these, for instance, the columns would be samples and the rows would be samples and the columns would be different genes. So we end up with regions of the genome, like for instance this white part here, where we don't have any power to deconvolve. So this is still under development. We've been looking at different ways to infer these values. I'm not going to talk about this today, but basically so far I'm staying with the median, but there's probably better ways to do that. So we validated CREDAC now, so this is without the inference, the actual value that you get out of CREDAC in using tumor and normal cell lines in silico mixtures. and also in the actual data using SNVs in regions with LOH. So basically, in those regions, we know that all the reads that have a mutation will come from tumor cells, whereas all the wild-type reads will come from the normal cells. So we can use this as a ground truth and compare it with our estimate of the convolution. And you can see here that we have good correlation with the ground truth, same in the cell line data. So what can we do with CREDAC once we've run it? So here's some of the examples of the things that we can start exploring. So this is, for instance, one sample, and now here each dot represents a gene for which we can obtain this expression per allele copy in the tumor versus normal cells. So we can start looking at genes that are expressed more in the tumor rather in the normal cells, and so on. We can also start looking at instead of looking at the gene level, look at the sample level. So on average, how much are the tumor cells expressing per allele copy than the normal cells? So here is what I'm plotting the median log-fold change between this ET and EN per sample, and you can see that it's higher than 1 for most of the cases. So basically, on average, the tumor cells are expressing more RNA-seq than the DNA cell per the same haploid copy, so to say. And then we can also start exploring different transcriptional dynamics, so different genes that, for instance, are expressed more in a tumor than normal cells can be involved, for instance, in cell cycle, DNA repair, or replication, whereas genes involved in the extracellular matrix organization or immune system are expressed more primarily to the normal cells. So yeah, with this as a conclusion, CREDAC is a purity and copy number aware bulk RNA-seq deconvolution tool. We output the expression profiles of the tumor and the normal cells, so just 2 compartments. We don't require the use of any reference signatures or normal samples, but we require matched DNA and RNA-seq data. And one of the limitations we're currently still working towards is that we're unable to deconvolve all of the genes in all of the samples. Ideally, if you have like cohort big enough, you would be able to find the estimate of all of the genes. So that's, in theory, you would be able to get them all. And then, yeah, so that's it. I just wanted to thank everyone in Peter and Charlie's group, as well as all of the TracerX patients and samples. And I also have a poster, so if you have more comments, suggestions, ideas, please come and say hi. Thank you.
Thank you very much. Last talk of the day. Any questions?
Curiosity. When you say normal here, it's not just a single population, right? Because most likely in your box there are many normals. And maybe some of them, they might have different expression profiles, you know?
Yes.
Doesn't this confound you?
So you will end up with like the normal and the tumor compartment, and it's true that within the normal, it's still many subgroups. So ideally, for instance, you can then apply other deconvolution tools to the normal compartment. No, I haven't tried that, but I want to try. But in theory, it shouldn't confound, since we're only separating like 2 dimensionals, everything that's aneuploid versus everything that's diploid. So it shouldn't be affecting that. But yeah, you can then couple CREDAC with our other deconvolution tools that are able to tease apart the normal compartment.
Any questions? Okay, then I would say we thank all the speakers of the session again. Before you clap and go, I would just like to ask you, all the speakers for tomorrow, please do send us your slides. So at the bottom of the program on the web page, there's the link. So do please send us, upload your slides so that tomorrow we have everything ready. Okay, with that, thank you.